I'm a neurology resident at the Erasmus Medical Center, but I also work as a postdoc researcher in the Alzheimer Center in Rotterdam, together with John van Sweden, and we prepared this presentation together, and he will be available for questions during the Q&A session later on. Well, we all know FTD is a very heterogeneous disorder, both with regards to the clinical phenotypes as well as the pathological substrates. What the central element of the disease is that there is degeneration of mainly the frontal and temporal parts of the brain. As you can see here, in a brain of a diseased patient where there's volume loss or atrophy in the frontal parts of the brain, whereas in the back side of the brain still a lot of brain volume, and this is visualized on this MRI scan on the right here as well. And we know that in FTD both the matter and the white matter get affected, and the gray matter are the neurons that are on the outside part of the brain, and in between is the white matter, which connects the different neurons with each other. And because the gray and white matter specifically degenerates in the frontal and temporal parts of the brain, the disease clinically presents with mainly behavioral disturbances and language difficulties. Most cases of FTD are sporadic, which means that it's not familial, but a smaller proportion of FTD is caused by genetic mutation, which can be passed on from parents to their children. And in this presentation, I will focus on that genetic type of FTD. So in order to do so, I will first explain some of the basics of genetics to you. Then I will give a short overview of the different genetic subtypes of FTD. And finally, I will discuss some of different research in genetic FTD and our plans for the future. So to start with the basics of genetics, each cell in our body contains our chromosomes, and every person has 3 different pairs of chromosomes, so 46 in total. 23 of the chromosomes you get from your father and 23 from your mother. All these chromosomes are built up of very long strands of DNA. And this DNA is built up of over 25,000 genes. And a gene is a small part of the DNA that has a specific function. And this function is determined based on their nucleotide bases. So the DNA is built up of these nucleotide bases, called, well, abbreviated with these letters A, T, C, and G. These bases determines the function of the protein. So you get a kind of letter code which is important for the function of the gene. So what happens? Mutation, this basically means that there's a change in that letter code. So here above in the right, you see an example of such a letter code. The letters are the nucleotides, and if there's a mutation, there's a change in this letter code. And there can be different kinds of mutation. You can see that one of the nucleotides is replaced by a different kind. Here there's an extra nucleotide inserted, and here one of the nucleotides deleted. And all these different alterations change the letter code, which may affect the function of the gene. In most cases, no effect of such a mutation at all. Sometimes there's even a beneficial effect of such a mutation. Happens in evolution, but occasionally such a mutation happens in such a crucial spot that it may lead to disease. That is because every gene is responsible for the production of certain proteins. So when this letter code is changed due to this may alter the production of this protein, which can either lead to too few production of protein or production of protein, and thereby it loses its normal function, or the protein can gain an extra function, which could be toxic for the body. And all these different changes in the protein production could cause disease, depending on the type of protein and its function. So as said, these mutations can be passed on from parents to their children. And in the case of mutations that can cause FTD, this happens in what we call an autosomal dominant mode of inheritance. This means patient has FTD due to a certain mutation, then their children have 50% risk to have this mutation as well and to disease as well. So this pedigree is an example. Here you see in orange the father who has one mutated gene, and this mutated gene causes the disease in his case. He married to a woman who doesn't have any affected genes, so she has two pairs of the gene. And when they have children, they both give one of their genes to their children. So in this, the left and the girl on the right both got the non-affected gene from their father, and of course also a non-affected gene from their mother. So they don't have a mutation, don't get the disease. But these two in the middle both got the mutated gene from their father, which means that they will also develop the disease. So now specifically for FTD, as I said before, FTD is usually a sporadic disease. So this means that one the disease and no other family members are affected. However, in 10 to 30%, there's a genetic mutation caused. We usually find such mutations in families where there are multiple affected relatives in multiple generations. But sometimes we find such a mutation in patients that seem sporadic because their parents are not affected or something. So it's always important to keep that in mind. The three major genetic defects that have been found to cause FTD are mutations in the so-called MAPT gene or the progranulin, and a repeat expansion in the C9orf72 gene. Besides these three, there are several genetic mutations. And I will briefly discuss the different genetic subtypes now. So the mutations in the MAPT gene were the first discovered genetic cause of FTD. And what we see when we look, we perform neuropathological examination on the brains of patients after they passed away from FTD, is when we look with a microscope, we see a so-called tau protein. So MAPT mutations are associated with a so-called FTLD tau pathology. Usually arise around the age of 45 to 65 years. And the symptoms usually include disinhibition or apathy, but a frequent clinical manifestation in line vertical basal syndrome, and some patients also suffer from epilepsy. This pedigree here is one of our large Dutch families, the MAPT mutations, and these lines represent the different generations, and all the black symbols are patients that were affected with FTD. These children are still at risk to also develop FTD. Then the mutations in the progranulin gene, they have a much wider range of onset age, ranging from about 35 to up to 90 years of age. Clinically, most also have a behavioral variant of FTD, but mostly with apathy and loss of initiative. Some patients have a non-fluent form of primary progressive aphasia. Here, some patients exhibit mostly Parkinsonism, like in corticobasal syndrome. In contrast to the MAPT, here neuropathological examination does not reveal the tau protein accumulation, but there's accumulation of the so-called TDP43 protein. We call FTLD TDP pathology. Here again is one of our large Dutch families with progranulin gene. So all these blue symbols represent persons that are affected by FTD due to the same progranulin mutation. Well, through many years, there have been many we found many different types of mutations in the same progranulin gene that can all cause. They all have in common is that when we look in the blood of these patients, there's a reduction of the so-called progranulin protein, and that's even in the pre-symptomatic stage. So before patients have symptoms, we already can measure this decreased progranulin protein. Then in 2011, there was a worldwide major breakthrough in the discovery of the repeat expansion in the C9orf72 gene as a major cause of both FTD, but also amyotrophic lateral sclerosis, or ALS, which is a disease that is characterized by muscular atrophy and weakness. And persons who have such a repeat expansion can either develop FTD or ALS or a combination of both. So in this pedigree, you can see that one family, so these are all siblings, and for instance, this person has FTD, this person has ALS, and this person has both and ALS. So there's a very large variation in clinical presentation. This clinical more pronounced because some patients mainly exhibit psychiatric disturbances such as visual hallucinations. Other persons have like Alzheimer mimic clinically with mainly memory disturbances. And there's also something that we call and Phenocopy FTD is a kind of benign form of FTD, which is characterized by relatively mild neuropsychiatric symptoms and cognitive decline, with a very slow progression over time. And also cognitive performance on neuropsychological investigation and brain volume measured with MRI is relatively stable over time. So often we possible diagnosis in these persons because progression over time is one of the key criteria for a diagnosis of FTD. So it's important that especially when such a person has a positive family history for FTD or ALS or psychiatric disorders, this could to repeat expansion in the C9orf72. So these different genetic subtypes differ to some degree in the clinical manifestation. To see a difference in the patterns of atrophy on MRI. So for the progranulin gene, we mostly see a rather asymmetrical, mostly affecting the frontal and temporal lobes. For C9 ORF72, the atrophy is more diffuse over the brain, and in MAPT mutation, bilateral temporal lobes that become affected. As said, there are also some less frequent genetic defects that can cause FTD, which are some examples are here. And for the Netherlands, especially the TARDBP seem relevant because we recently found a relatively high frequency of these mutations in familial FTD in the Netherlands, which was higher than in although all here is a pedigree of this Dutch family, and although all these persons came to us individually without a known family relation between them, through genealogical research, we found that they are all related. An ancestor possibly many years ago. So it's basically a very large family. And clinically, these mutations with mainly behavioral disturbances and semantic deficits. Then the research in genetic FTD. Why do we do this? Of course, it's important to discover these genetic mutations for appropriate genetic counseling in the clinical practice, but also these genetic subtypes of us a lot about the underlying disease mechanism, from how the genetic mutation can finally cause the disease, which is crucial for the development of disease modification. And finally, it can really help us to develop appropriate biomarkers for the earlier stages of FTD. The FTD risk study, which is a study where we ask healthy family members of patients with FTD due to one of the known mutations to participate, and all these participants have 50% risk to carry such a mutation and thus to develop FTD in the future. They have no symptoms at all. We analyze with different investigations, including MRI scans and different measurements in blood and cerebrospinal fluid. So we study in 2010, and now we have over 200 participants. And of course, during follow-up, some of the persons with a mutation will develop symptoms of, and this is what we call the converters. So they convert to the symptomatic stage of FTD. And as the UCL group already mentioned, there's now also collaborative with basically the same aim, where we investigate both the symptomatic and the pre-symptomatic stage of FTD, and all the risk study are also included in this GEMFI initiative. So this slide shows one of the first of our first things in the FTD risk study. Here we compared carriers of the mutation with non-carriers, and again, all these persons had no symptoms yet. And then looking at the gray matter volume, we found no differences between the groups. But when looking at the white matter integrity, we saw increased white matter integrity in this structure. So this means that changes in the brain can already be measured before symptoms of FTD. And of course, we followed these individuals over time. And after 4 years, 8 persons with a mutation had developed symptoms of FTD. So this is what we call the converters. And we compared them with the mutation carriers that did not develop symptoms yet. And what we saw is that at baseline, there were no differences between the groups at all in either the white matter integrity or the gray matter volume. But later, so still 2 years before their symptom onset, we see large differences in both the white matter integrity and the gray matter volume, which is even more pronounced time of symptom onset. So this means that there's a rather explosive gray and white matter disruption relatively short before symptom onset. Besides these MRI measures, we also looked at several biomarkers in blood and cerebrospinal fluid, and the Neurofilament is an important one. This is a rather aspecific marker for loss of neurons, and it was shown to be at the symptomatic stage different genetic subtypes of FTD. And more importantly, it was shown to correlate with both measures for cognitive performance and with brain volume. We that this might be a useful measure to predict disease severity and progression. A different kind of protein we looked at is the neuronal pentraxin receptor protein in cerebrospinal fluid, which was shown to be lower in the symptomatic stage of the genetic subtypes. Also correlates with brain volume in the frontal lobe. And when looking at it longitudinally, then it's to see that around the time of symptom onset, there's a really prominent decrease in this protein. And therefore, we think that this protein might be valuable to predict symptom onset. And we see an even further decrease during the symptomatic stage of the disease. So this was, in a nutshell, the research we've already done in genetic FTD, and our major next focus is to look at the role of neuroinflammation. Over recent years, we have seen that besides degeneration of the brain, there's also a kind of inflammatory process, but we don't know what the exact role is in the disease process. And unraveling this role is important to further understand the disease, but also to see whether anti-inflammatory agents could be a useful therapy for FTD. And finally, we also, previous research has also shown that some for neuroinflammation, like in blood or cerebrospinal fluid, but also imaging markers with MRI or PET, could be helpful to predict disease onset in FTD, track progression. So with our upcoming research, we want to further investigate this role of neuroinflammation using these biomarkers, but also with studies of neuroinflammatory cells and with animal models. So that's what we will do. And then important for the progranulin subtype of FTD is that there's a trial coming up, especially for progranulin carriers, and you will hear more about this at the talk at 2 p.m. So this is what I wanted to tell you. Thank you all for your attention, and I'll now give the to Hendrik Jan.